Risk assessment or risk management in clinical laboratory. Uh, why this topic has come suddenly since last 2 3 years, everybody is speaking about risk management in clinical laboratory. This is not something a new topic which we have been talking since long, but suddenly during the COVID season or during the COVID pandemic, suddenly the risk assessment in clinical laboratory has come up, and it has been emphasized a lot as far as the medical laboratories and clinical laboratories are concerned. First of all, let me tell you, this is a risk awareness week is going. It is celebrated almost on the third week of October throughout the world. So this year it is from 17th October to 21st October 2022. The risk awareness week is being celebrated throughout the world. So I am very glad to speak on the same topic in this occasion. So basically, in this presentation, I'll be covering about the what is risk and risk management. Then we will see about the actual process of risk management. So there are certain stages or certain steps which we have to follow during the risk assessment and risk management. And finally, since this is a slightly a theoretical topic, I have taken some example or maybe a case study, you can say, at the end, so that we can properly understand what is in by risk management and how you can apply it in your laboratory. So to begin with, I have put first of all this fellow. So maybe most of us will be knowing this is from the series Scan 1992 about the big bull, and here it is written risk hai to ishq hai. So basically that means there is a risk in love. So basically not only in love, the risk is everywhere, and the risk is limited not only to the laboratory, it is related with the finances, it is related with the stock market, and even if you see in the commercial advertisement of mutual funds, it is mentioned that mutual funds investment are subject to market risk. So basically risk is involved everywhere, and there must be a process to eliminate this risk or to minimize the risk. So here, if you consider a stock market also, if there are investment at this level, there are chances of getting a high return, and there are again chances of losing your money. So basically there is a risk to your wealth. So this is the most common example what practically we come across as the risk of the financial wealth or the risk in the wealth in the stock market. So what is the risk and risk management? So basically risk is the something happening bad or something which is there is a probability of something which is going to be a wrong. So that is called a risk. So risk is something which is going to be wrong, and risk management is basically we can say it is these are the coordinated efforts where which we can try to minimize or we can try to eliminate the risk. So risk is basically the probability of harm or the probability of anything going wrong, and risk management is to remove this probability or to remove this harm or minimize the harm in the risk management process. So as I have told, the concept of risk is not new. For the years, gamblers, investors, financials, manufacturing sectors, service sectors, transportation sectors, airlines, so there is always a risk. So there is always a risk, and there is always a probability of harm on all these sectors. So recently it has been introduced to the healthcare sector. So the concept is not new. The only thing is it is just new to us. So in healthcare sector, the risk concept has been introduced in 1970, whereas in patient safety program, the risk concept was introduced in 2000, and for medical laboratories, the concept of risk and risk management has come in 2003. So we are going to see the concept of risk management in medical laboratories. So how this concept has evolved over a period of time as a part of quality management system. Sorry. So if we consider the evolution of quality management system over a period of time, you can see, first of all, we were looking into the quality control measures, then it came for quality assurance, then we have started looking into the quality systems, then we started looking into the overall management and environmental management of the laboratory, then there was a quality management system completely, then there were occupational safety management systems, then there were total quality management, and now what we are looking is the risk management. Even after the risk management, or even if you go beyond, there is process modeling and integrated management system. So we are right now, we are in this stage of risk management system, and many places, if you see, we have already moved to the process modeling or integrated management system. So this is how the process of quality management system has evolved over a period of time, so that the risk management systems we are now actually considering in our laboratory. So why to manage a risk in clinical laboratory? Basically, why we want to manage this risk? What is the requirement, or what are the necessity of managing the risk in clinical laboratory? So to consider, here I have put two images. You can see at the bottom, this is one image which shows about the modern days manufacturing units or modern days factories which are fully automated and multiple processes are going at a time. So similarly, if you see this picture, this is basically a picture of a modern day clinical laboratory. So if you can consider, the modern days laboratories can be directly compared with the modern days manufacturing units which are into the manufacturing sectors and which are manufacturing the product. So similarly here also, we are not manufacturing any product, but our products are basically the test results. So if you consider this modern days laboratories, there are multiple processes, there is an interaction of these multiple processes. So there is a high chance of getting harm or going something wrong, which can cause a risk to the processes in the laboratory, or which can damage the process in the clinical laboratory. So all these processes are interrelated with each other, and here for this purpose I have simply put one fishbone diagram. What is fishbone diagram has been already explained to you by Dr. Mahesh, so I am not going into the details. So but to consider that to get the incorrect results, there are multiple factors and multiple variables which can affect the test results, and all these variables poses a harm, or all these variables can form a potential risk if they are not in the appropriate process, or if they are not followed appropriately in the specified manner. So all these variables becomes the part of risk. So we can see here there are multiple variables and multiple risk factors which can affect the risk management or risk in the clinical laboratory. So now why this, what this risk will have effect? This risk have various effect in the healthcare sector. So the footfalls of the risk in the medical laboratory is not limited only to the laboratory staff, or not only related with the patients, but it can affect the clinical staff, it can affect the complete institution, it can affect the community and environment. So if the biomedical waste or any other hazardous materials, if they are not disposed of properly, they can cause a risk to the environment as well as the community. So the footfalls of the risk in the clinical laboratories are to the patient, to the clinical staff, to the laboratory staff, to the institutions or to the organizations where the clinical laboratory is functioning, to the community where the clinical laboratory is working or operating, and to the environment. So basically there are multiple factors and the multiple footfalls for which the risk can affect, from the risk in the medical laboratory can affect this all the areas. So it is very important to manage or to nullify these risks which are involved in the medical laboratories. So how this risk management process is done? So basically here I have given one diagrammatic representation where you can see the complete life cycle of risk management process. So first of all, if you see these small rectangles in the center with the broken lines, here there is a step of risk analysis. The outer rectangle, if you see, it mentions the risk assessment, and the overall complete rectangle shows the complete life cycle of risk management. So the risk analysis start with the hazard identification or to identify the potential risk. Then you have to consider the severity of the risk, probability of the risk or probability of the harm. You have to estimate the overall risk, and then you have to evaluate it, how much it can affect your processes or how much it can affect your other processes in the laboratory or to the other footfalls of the laboratory. So with this you need to perform the risk prevention and control measures, which can again consider after performing this risk assessment prevention control measure, again you can have a risk analysis step, and if they are falling into place, you have to control the risk, and then you can monitor the risk over a period of time. So if the risk control measures are working appropriately, you need not have to do any further measures. But in case there are any failures, or if there are any more risk involved, or if there are any new hazards, you need to perform a complete risk analysis and complete risk assessment process. So this is the complete life cycle of risk management process in a clinical laboratory. So what are the important documents which we are going to refer for performing the risk management? So there are various documents and international standards which are used for the risk management processes. So first of all is the ISO 22367 document 2020 editions, which mentions about the application of risk management which is specific to medical laboratories. There is one more standard, ISO 31000 2018 edition, which deals with the risk management, a practical guide. ISO 31010 document 2019 edition, which mention about the risk management techniques. So there are various techniques basically which are involved in risk assessment and risk management. So this document deals with those techniques that are involved in the risk assessment and risk management. Then there is the ISO 14971 document 2019 edition, which specifically deals with the risk management for medical devices. And finally there is a CLSI EP23A guidelines, which specifically deals with the laboratory quality control based on risk management. So these are the some of the reference documents which are used for the risk management or the risk assessment in clinical laboratory. And of course our ISO 15189 2012 document, which also mentions about the risk management and which is specific for the medical laboratories. That is also one of the document which can be used for the risk assessment of medical laboratories. So these are all documents which can be used as a base or a referral document for the process of risk management. Now what are the steps or what are the sub processes that are involved in the risk management? So the risk management process starts basically with the planning. So first is the risk management process planning. The next is the risk identification or hazard identification. After that, risk analysis and evaluation. Next step is the development of risk response plan, and finally the risk monitoring, controls and revisions. Revisions will be required if there are any more risk or if the severity or hazards of the risk are not controlled, then you may need to revise the plan and you can have a new plan for the risk management. So to start with the planning, so risk management process planning, first of all you need to define the scope. So we know there are various areas in the medical laboratory that starts with the pre-examination, post-examination and examination areas. Then there are certain managerial areas, supportive areas. So first of all you need to define an appropriate scope. So to which area the risk management will be applicable or which areas you are going to consider for the risk management. So first of all you need to define the scope of your risk management process. The next thing is assign responsibilities and authorities. So you need to assign one person or a team for this risk management process. So you can have a risk management team where you can have a risk manager, you can have a quality manager who can take care of the risk management process, or you can have a lab director himself who can take care of this risk management process. So he should have a responsibilities. He or she who is the dedicated person must have responsibilities for the risk management process, as well as that person must be provided with the authorities to take decisions and to make changes in the processes. So you need to have assigned responsibilities and authorities for the risk management. Next step is the defining the criteria for risk acceptability. Now, once you have evaluated the risk in your laboratory processes, you need to find out what are the criteria for risk acceptability. How much risk is acceptable and how much risk is not acceptable, or how much risk you need to have appropriate corrective actions or process modification. So first of all, you need to define your criteria for risk acceptability. So since you are making a plan, all of the things should be defined in advance. So basically you should have an appropriate process in place for the risk management. So there itself you have to define your criteria for risk acceptability. The next thing is specify your verifications, risk monitoring and documentation requirements. So in a risk management process, how you are going to verify the process, or how you are going to verify the control measures. So whatever control measures you have taken for removing the risk or for elimination of the risk, you need to specify the criteria for verifications. When you have implemented the risk management process or risk control measures in your laboratory, you need to specify the verifications, or you need to have a appropriate verification of the risk control measures. And then you have to define the risk monitoring measures, how you are going to monitor the risk, or how you are going to monitor the hazards in the processes. And finally, what are the documentation requirements? So everything comes with a documentation. Unless and until it is documented, it is not done. So you need to have a appropriate documentation process. So whatever actions you have taken, whatever measures you have taken to control the risk, and what are their effects, how much the risk you are able to eliminate. So you should have a appropriate documentation of this risk management process. So first of all, in the risk management, you should have a appropriate planning where you are going to define the scope, assign responsibilities and authorities, define criteria for risk acceptability, specify verifications, risk monitoring and documentation requirement. Now the next step is basically the risk identification. Risk identification is basically nothing but identification of the hazards, or identification of the possible risk or possible hazard which can affect the complete scenario or complete test results, which can affect the patient care or healthcare delivery. So you need to identify such risk areas or such harm in your laboratory. Now which areas you are going to look for risk identification? As I have already mentioned, there are areas of pre-examination, examination and post-examination. You can also consider some of the managerial areas or administrative areas or supportive areas such as purchase department, how much or how they are selecting the equipments, how they are selecting the consumables, how much their quality can affect the risk, or how much their quality can affect the patient test report. So you need to have a appropriate risk identification areas from the pre-examination, examination and post-examination activities. You can also consider the areas of housekeeping, where a simple dust can also affect the functioning of your equipment and which can cause a inappropriate or invalid result. So you need to first of all define the risk, or you need to identify the risk in all these area. Now how to identify the risk? So basically there are certain tools and techniques. So what are these tools and techniques? So I will just mention here a list of these tools and techniques. So I think most of them will be familiar to you. Primary hazard analysis, failure mode effect analysis or FMEA analysis. Then there is a fishbone diagram, there is a process mapping and fault tree analysis. So what is meant by fishbone diagram is already discussed to you in the previous lecture by Dr. Mahesh. These are the other techniques and tools which can be used for the risk identification in various areas of the laboratory. Since the scope of this presentation is limited, I am not going to into the details of all these tools and techniques, because these are the general tools and techniques which are used in the quality management system. So once the risk is identified, you need to analyze the risk and evaluate the risk. Now the risk analysis and evaluation is considered under two different headings. One is the risk acceptability criteria and the second one is the risk evaluation process. So the risk analysis and evaluation is considered under two separate criteria of risk acceptability criteria and risk evaluation process. Now where is this risk acceptability criteria? If you remember, when we are doing planning of the risk management process, that is the first step of the risk management, we have done we have defined our risk acceptability criteria. So this risk acceptability criteria can be determined according to the laboratory's policy for determining the risk acceptability criteria. So these criteria we have already defined in the first step where we were defining the process of risk management. So the risk acceptability criteria are already defined in our process. It can be based on laboratory's own policies. It can be based on applicable national or regional regulations, applicable safety standards, relevant medical practices standards, can be taken into account general acceptable state and art of stakeholders concerned, and it can be approved by the laboratory director. So what are your risk acceptability criteria? It can be laboratory's own policy, which can be based on national and regional regulations. You can also take help of some peer review journals, standard textbook, or you can have also some discussions among the experts in the field, so as you can define the risk acceptability criteria. And all these risk acceptability criteria must be approved by the laboratory director. So while evaluating a risk, you must have a risk acceptability criteria. How much risk is acceptable for any of the process in medical laboratory. Now the next step in the risk analysis is the risk evaluation process. So in risk evaluation process, again there are three steps. One is the probability of harm, severity of the harm, and detectability of the harm. So here we have some scoring system based on which we are going to evaluate the risk. So the scoring system based on three variables, that is the probability of harm. So what is the probability that a harm can occur? So in a process, if anything goes wrong, what is the probability of harm, or what is the probability, or what is the chances of the getting the harm? Then once there is a probability of harm is detected, you need to find out the what is the severity of harm, how much severe the harm can be, or how much severe the hazard can be, whether it can be life threatening or whether it can be a trivial harm. So you need to define the severity of the harm and the detectability of the harm. So in many a time the processes itself have a their inbuilt system in place where the harm can be detected by the process itself. So once the harm is detectable by the process itself, the harm can be removed by the process in the process itself. So you need to consider all these 3 points while evaluating the risk. So based on these three things, we are going to put a scoring system. So first of all, we will see the probability of harm. So probability of harm, so there are 5 levels: level 1, level 2, level 3, level 4 and level 5. So if the probability of harm is very frequent, or the probability of harm, or if the hazard can occur on every day or on day-to-day basis, you can call it as a frequent hazard or frequent probability, and you can score it as a 5 points. The next is the hazard can occur on weekly basis or maybe once in a week on an average, so you can score it as a 4. If it is occurring once in a month, you can score it as a 3. If it can occur once in a year, it can be scored as a 2. And if it is less than once in a year, or it is very unlikely that the harm can occur, you can just measure a level 1. So please remember that there is no zero scoring at any place. So there is no zero score, or there is no level 0 harm for any of the risk involved, or any of the probability of harm in the risk management process. So if it is very frequent, we are going to score it as a 5. If it is very unlikely, we are going to score it as a 1. So the probability of harm is scored from 1 to 5 on the basis of its probability or on the basis of its frequency. The next is the severity. So we have to define how severe is the harm. So again here we have scored it from score 5 to 1. If it is a very critical or catastrophic, we are going to make it or score it as a 5. If it is a life-threatening effect or injury or death can occur, it is a critical or catastrophic event, we can score it as a 5. If it can cause permanent damage or irreversible damage or impairment, it can be categorized as serious issue or serious harm. So we are going to score it a lot of score of 4. If it is a significant harm which can cause non-permanent bodily damage or impairment reversible with the medical intervention, we can score it as a 3. If it is a marginal category which can affect temporary bodily damage or impairment which can reversible with no medical intervention. So there is a difference between the score 3 and score 2, where score 3 the impairment can be reversible with medical intervention, and score 2 is the reversible with no medical intervention. And last category is negligible harm, where the score is 1 and there will be no there can be a simple temporary discomfort or inconsequential injury. So here again the severity we have scored from 5 to 1 on the basis of its severity from critical to negligible. Now we have to see the detectability of the harm or the detectability of the risk in the process. So again here again we have scored values from 1 to 9, where there is a high degree of detectability. So if the harm can be detected by the process itself, we are going to assign a score of 1. If it is a good detectability, we are going to assign a score of 3. If it is likely to detect, we are going to assign a score of 5. If it is fairly detectable, we are going to assign a score of 7. And if it is low or no detectability, we are going to assign a score of 9. So based on all these criteria such as severity, probability and detectability of the harm, we are going to evaluate the risk. Now how this risk is evaluated? So it is evaluated on the basis of RPN, that is risk priority number. So this risk priority number takes into consideration of probability of harm, which we have seen scoring from 5 to 1. Then we have seen severity of the harm, where we have again seen the scoring of from 5 to 1, and detectability of the harm, where we have seen the scoring of 1 to 9. So to see the maximum RPN which is possible for a process will be a maximum probability score can be 5, maximum severity score can be 5, and maximum detectability score is 9. So considering the all these 3, 5 into 9, 125 is the your maximum score. So any process or any harm can have a maximum risk priority number is 125. Now what is the use of this risk priority number? As the name suggests, risk priority number is based on these numbers or based on this scoring system, you have to prioritize the risk, or you have to prioritize the risk hazard. As we have seen in various areas, we are going to have a risk evaluation process. There will be multiple hazards. So on these multiple hazards, some will have less probability, some may have more probability, some may have less severity, some may have more severity, some may be detectable and some may not be detectable. So on the basis of this, we are going to prioritize the risk. So which risk is on our priority basis. So on the basis of that, we are going to take first action, or we are going to the risk assessment on that area. So first of all, you need to prioritize your risk. On the basis of the risk, you are going to manage the risk in your laboratory. So that is why we are going to use this risk priority number. The maximum risk is 125, and minimum, as I have told, minimum score for probability is 1, minimum score for severity is 1, minimum score for detectability is 1. So 1 into 1 into 1 will be 1. So lowest probability for risk priority number or lowest risk priority number will be 1. 1 will be considered as a least risk areas, and 125 will be considered as a highly risk or highest risk area. So based on this scoring system, we have to prioritize your risk management. Now, once the risk evaluation is done on the basis of risk priority numbers, you have to control those risks. So how to control these risks? First of all, you have to identify ways to reduce those risks. How you can reduce these risks? You have to prioritize the risk reduction measures. So there will be multiple measures. So once you find out the risk in your area, and once you have defined that which risk you are going to first address, you need to prioritize measures. Now how you are going to prioritize these measures? So again, for this purpose, there is one more tool which is called as Pareto chart. Again, this chart has been already discussed by Dr. Mahesh in the previous lecture. We have to focus on the vital areas, and this Pareto chart basically it shows about the 20 vital areas and 80 tribal areas. So you have to focus on these 20 vital areas where you can have a maximum impact of the risk management, or you can have maximum impact for the risk control. So that is how you can prioritize the risk management control or risk control measures in your laboratory. So you have to focus on those areas which are vital and which can be more useful for reducing the risk, rather than focusing on smaller areas, or rather than focusing on trivial issues which will be multiple in numbers, but controlling those issues may not solve the problem in your laboratories. So first of all, you need to focus on these vital areas where you can have a appropriate risk control measure in your laboratory. So the next is the risk controls. What are the different approaches which can be used for the risk control? So as I had told, inherent safety by process designing. Many of the processes will have inherent safety process. So they will have the measures in the process itself where the risk can be controlled by the process design itself. So the process itself is designed in such a way that the risk can be controlled by the inherent or inbuilt safety measures. Next category of risk control measures may be use of protective measures in IVD medical devices, or in the examination, pre-examination, post-examination areas. Quality assurance procedures you can have, or you can have rescheduling of quality assurance measure, quality control measures, calibrations, quality control activities, including new control activities which can be added to reduce the risk. Information to the staff for the safety. So appropriate safety measures should be taken care by the staff, and most important is the training of the staff or training of the personnel who are involved in the a specific process. So the risk control measures can be various measures. The most important is the training. Sometimes there can be inherent safety measures in the process itself which can help in reducing the risk, or which can help in controlling the risk. Other areas can be emphasized by just following the IVD medical devices instructions which are given, which includes alarms, failure detection, fail-safe mechanisms, and other quality control measures of pre-examination, examination, and post-examination, implementing appropriate quality assurance procedures, appropriate calibrations, appropriate equipment maintenance, and training of the staff. So basically you need to have appropriate risk control measures which can help in reducing the risk, or which can help in eliminating the risk. So once the risk control is done, you should have a risk management review. So you need to review the process. So once the control measures are in process, you need to again review the complete process. So you have to review the process where the risk has been identified. So for that purpose, you need to see the completeness of risk control. So whatever control measures are put in place, whether they are complete or not, whether they are sufficient or not. Once the risk control measures are put in place, whether there are any residual risks, so you have to evaluate for overall residual risk. So even after putting control measures in place, whether there is any residual risk, that you have to find out. And finally is the risk management report. So you have to make a proper risk management report which will start from the risk planning, and step by step risk management process in your laboratory. So a complete report has to be prepared, and which can include the completeness of risk control as well as any other residual risk is remain in the process or not. So a complete report needs to be prepared for the risk management review. And finally, the risk monitoring, analysis and control activities. So once the risk management process in place, control measures are taken, the next step is you have to monitor the risk. So you have to monitor for long term basis that whether the risk control measures which are put in place to control the risk, whether how much the effectiveness of these risk control measures, and you have to analyze the control activities, and if there are any more issues or if there are any more risk or hazards involved in the process, those needs to be addressed with the different processes or different process designs. So one more aspect here I am covering is the basically the risk matrix, how to plot a risk matrix for the risk management process. So you need to classify it under 5 zones. This is a sample risk matrix I have put, where the risk severity and probability is plotted against the detectability. So here on the y-axis we have plotted the risk severity and the probability. So it will be a multiplication of risk severity and probability, and on x-axis the detectability. So here you can see the 5 zones. One is the green, dark green zone, then there is a light green zone, then there is a yellow zone, orange zone and red zone. So what these 5 zones shows, so these 5 zones, the light green, the dark green shows no risk, where no action will be required. The light green zone will show you small risk where appropriate training can be done to remove the risk. The yellow zone will show a moderate risk where the cost effective and selective controls can be taken up. The orange zone shows an unacceptable risk where the controls irrespective of the cost involved must be taken care, and the red zone is the severe risk where immediate change of process or design and control measures are required. So on the basis of this risk matrix, you need to classify the risk priority using the risk matrix. So finally, I have included one case study just to have a small idea about how to practically implement this risk management in clinical laboratory. So for considering this, I have considered the area of pre-examination area in the medical laboratory, where I have considered the phlebotomy procedure. So in phlebotomy procedure, again has lot of small processes in which I have considered the process of patient identification. So patient identification is basically, as per the international guidelines, you should have 2 positive patient identification at least. So positive patient identification include the patient should be able to tell his name appropriately, and it should be matched with his unique identity number. So these are called the positive patient identifications. You need not have to ask any leading questions, or you need not have to ask a yes no type question. You need to ask the patient his name and his other details by himself by communicating to the patient, and it should be matched with the unique identity of the patient. So in pre-examination area for the phlebotomy procedure, we have considered the process of patient identification. Now what can go wrong here if the patient identification is not done properly? There can be a wrong patient identification, and there can be mislabeling of the samples or mismatch sample. So the hazard involved here is the mislabeling or mismatch sample. Now the probability of this is once in a month. Now how I can say it is once in a month? I can say this on the basis of data which I have obtained already from my laboratory. My laboratory means here I am giving it just for example. The probability of occurring this event of wrong patient identification is a once in a month process. So this I have to, for this purpose I need to have a complete data of wrong patient identification which is occurring in my laboratory. On the basis of that, I can define that the probability is on an average once in a month I have the evidences or I have the incidences of wrong patient identification. So the probability of occurring a wrong patient identification is once in a month for my laboratory. Now what is the, as per the scoring system, the category comes with the score which we are going to allot is 3. So the probability of occurring harm is occasional and the score which we are going to allot is 3. Now what is the severity of harm? So wrong patient identification is always a critical event, or it is a severe event which can cause a mismatch processing of the samples and which can lead to a wrong report to the patient and which can cause a wrong treatment or inappropriate treatment to the patient, which may or may not be a appropriate for the patient, or which may or may not be causing damage to the patient. But since there is a wrong patient identification, as per the international classification, the severity of identifying a patient wrongly is always a critical event. So the score again I have allotted here is a 5. So now the probability score is 3, severity score is 5. Now my third aspect is detectability. So here detectability I have mentioned as a likely to detect. Now why I have mentioned it is likely to detect? Because once the patient is wrongly identified, the sample is sent to the analyzer after appropriate sample preparation. Now the sample, once it goes to the analyzer, if the analyzer has already processed the samples with the same identity, in that on that day it may not process the sample again. So if the patient is labeled with any wrong patient identification, or here we can say any wrong barcode, if the barcode is already read by the analyzer for some test, and if a wrong barcode is pasted on the sample and the sample is introduced in the analyzer, the analyzer may not run the samples, or the analyzer may decline the processing of the sample based on the inbuilt ability of the process. So inbuilt ability of the laboratory information system, or LIS, it will detect that this sample is already processed and it may not process the sample. But the detectability of this is likely to detect if the process has some measures in its place. So that is why we have label it as a likely to detect. Since if there is no measures, or if the LIS is not capable of detecting such errors, it may go undetected and the samples will be tested as a wrong samples. So that is why here the detectability is calculated or is assigned as likely to detect, and the score allotted is 5. So now I have a probability of 5 for the event, severity of the event or the harm is 5, and the detectability which is likely to detect is also a score of 5. So here my RPN, that is risk priority number, is 5 into 3 into 5, which will come as a 75. So now my risk priority number is 75. Now whether this 75 score I should take appropriate action on this risk area, whether I should try to eliminate this risk or whether I should keep it as it is, whether this severity of this event is significant for my laboratory or not, this I have to define. As we have told in the first step itself, we have to define the acceptability of the risk. So here the RPN is 75. Now I have already defined that RPN 75 whether it is acceptable or not. So for me it is not acceptable, and the risk management action plan in my category, it comes as an unacceptable risk in a zone 4 of X4, which mentions that controls should be done irrespective of the cost involved. So here the RPN is scored on the basis of zone. So this was my plan when I have performed the planning for risk management in my laboratory. RPN less than 75, I am not going to do any actions because there is no risk. RPN 25 to 49, there is a small risk involved, so I am going to do a small training programs which can eliminate the risk. RPN 50 to 74, there is a moderate risk. So the cost effective measures and selective measures I have to do for controlling the risk. RPN 75 to 99, it is an unacceptable risk and I have to control the risk irrespective of the cost involved. And RPN 100 to 125 is the severe risk where I need to do immediate changes of the process or design and control measures. So in this example, the RPN is 75. So for me, it is an unacceptable risk and I have to take measures irrespective of the cost involved. So this is how appropriate risk management is planned in a laboratory. So with this example or with this case study, I have explained the how to perform a risk management in your laboratory. So after having appropriate measures at its place, now once I find out or once I have taken measures to control the risk, now I have to find out what are the residual risks or evaluations. So even after having my control measures in place, I need to evaluate the residual risk. So this residual risk will be evaluated, and again the severity, probability and detectability of the residual risk will be evaluated. RPN will be calculated again for the residual risk, and depending upon the RPN, again it will be determined whether the risk is acceptable or unacceptable. If the risk is again acceptable, I can continue my processes in the same way. If the risk is not acceptable, again I have to do a control measures for removing the residual risk. So with this, the short summary of this presentation is basically risk management can minimize the chance of errors and ensure reliability of your test results. Risk management guidelines recommends that laboratory play a proactive role in minimizing the potential for errors by developing individualized quality control plans and your quality control processes to address the specific risk encountered with the laboratory analysis and laboratory processes. So that is how we plan to do a risk management or risk assessment in our laboratory. With this, I will conclude this session. Thank you.